Good day everyone, I am Roxanne Siroas, a 4th year student, and I will be your teacher for today.
Okay, before we will start our topic, allow me to introduce the learning outcomes first. At the end of this lesson, the learner should be able to, first, define what a gene is, second, differentiate gene therapy from stem cell therapy, third, explain the importance of gene therapy in the future, fourth, demonstrate processes of gene therapy, and lastly, show in life situations genes' importance in education.
I know that some of you here are familiar with gene therapy. Allow me to take a review on what gene therapy is. So, what exactly is gene therapy? First, let's start with the basics. This is a gene. Genes are made up of DNA, which are the blueprints to build the enzymes or proteins that do the work for your body. As far as we know, humans have between 20,000 and 25,000 genes, and most people will have 2 copies of each gene inherited from each of our parents. They are the building blocks of our physical makeup, from the color of our hair, to our athletic abilities, to our immune system. But sometimes genes are built correctly, and a small adjustment to DNA within our genes can change how our proteins work, which can affect how we breathe, walk, or digest food properly. If the genes are like the blueprints of our body, gene therapy can fill in some missing drives. Gene therapy is the introduction, removal, or change in genetic material, specifically DNA or its molecule causing RNA, into the cells of the patient to repair or change the gene to treat a specific disease. The transferred genetic material then changes how the protein of group of proteins is produced by the cell. It can reduce levels of soothing disease-causing proteins, increase production of disease-fighting proteins, or even produce new or modified proteins within the cell. You may be curious how a gene and the genetic material it holds is transferred to the cell during gene therapy. Well, a gene cannot be directly inserted into a cell. Instead, the carrier, also known as vector, helps deliver the gene. Typically, viruses are used as vectors because they are evolved to be very good at sneaking into and affecting cells. But in this case, their moving is into insert the gene or genes into the cell. The infectious part of the virus is removed, allowing it to still enter the cell nucleus without making the patient sick. It's worth noting that there are 2 kinds of gene therapy, in vivo and ex vivo. In vivo gene therapy, the genes are introduced directly into the patient's cell while the cells are still in the patient's body. In ex vivo gene therapy, cells are removed from the patient's body, then the genes are added to the cells in the lab, and cells are then returned to the patients. The right approach depends on the disease that the patient has and the tissue that affected, and that will tell us which is the best way to approach this.
Genetic risks to health come from a variety of sources, and predicting those risks can be much more complicated than simply assessing diseases' inheritance patterns. The majority of health conditions are not related to a single genetic cause, but result from a combination of inherited and environmental influence. Because chromosomes and genes provide instructions for our bodies, any changes to these structures have the potential to cause genetic disease and negatively impact health. Heredity is the passing on of traits from parents to their offspring, either through asexual reproduction or sexual reproduction. The offspring cells or organisms acquire the genetic information of their parents through heredity. Variations between individuals can accumulate and cause species to evolve by natural
Selection. Let's move on to
understanding gene therapy. The expression of genetic conditions is not always as simple as the amount of DNA or the deoxribonucleic acid present or the inheritance patterns of a single gene disorder. There are many factors that determine how one's genotype or the genetic makeup correlates to one's phenotype or the physical manifestations. Among these are penetrance, expressivity, mosaicism, and X inactivation. Penetrance is a measure of how often a disease genotype correlates to the disease phenotype. For example, In a completely penetrant disease, every individual who is genetically positive for the disease would have clinical manifestations of said disease. In contrast, for diseases with incomplete penetrance, some individuals who are genetically positive for the disease may be entirely asymptomatic. Organisms inherit genetic information in a variety of ways that result in continuity of structure and function between parents and offspring. Suppose you as a child have a genetic disease, one that could be cured by diet or a similarly straightforward therapy. Wouldn't the parents want the disease diagnosed? Or take another example. Perhaps two people are getting married. Wouldn't it be beneficial to know what chance their offspring have of inheriting one or several harmful genetic traits? We are now armed with the different advanced procedures like the PCR, Southern blotting, RFLP mapping, and DNA sequencing. Different laboratories have already developed the ability to provide a very detailed look at our genetic materials. Is there a necessity to apply all these techniques to all of us? What harm might it do to have a complete picture of our genes? In totality, these procedures do not insert new genetic material into cells. No changes of any kind are made. We are only provided with information about what is already there. To further understand, please watch this video.
Hi, I'm Jean, and I'm here to help you understand what gene therapy is. But first, we need to learn about genes. Genes are the blueprint or instruction manual of all living things and can be found in each and every one of your cells. Using this blueprint, cells are able to get to work. Genes affect everything from the color and texture of your hair, to your height, to how your body metabolizes protein in the food you eat. Genes are made from your DNA and carry your genetic information. Sometimes there is a change, also known as a mutation in the gene from the original copy. This mutation or defect in the gene causes it not to work properly. Mutations can come from biochemical processes within the cell, something outside of the cell, or what we inherit from our parents. Genes that aren't working properly can have serious consequences. Scientists are working to find ways to solve this problem. They can replace, add, or turn on or off genes to tackle many health issues. PKU is caused by a mutation in the PAH gene. Because of this mutation, people with PKU cannot metabolize the amino acid phenylalanine, or as we call it, Phe. Phe comes from protein in our diet and has many important jobs, including helping our brain function normally. If the body cannot properly process Phe, the Phe builds up and acts like a poison that can cause a number of brain problems, including PKU fog. In gene therapy, scientists can repair a defective gene, add a new gene to fix the problem, or even turn off a problem gene. For PKU, scientists are working on introducing a brand new PAH gene. The PAH gene is made outside the body in a lab, and then it's transferred into the body to replace the mutated PAH gene. This new PAH gene will tell the cells to make the protein that will process Phe and help protect the brain. But to get into the cell, the new gene needs to first hitch a ride. Scientists use viruses. These viruses are called vectors, which are like little delivery trucks to get the repaired gene into your body. But don't worry, scientists make a virus safe by removing all the stuff that can make you sick. Then the delivery truck, also known as vector, carries the repaired gene into the cell. Once the gene moves into its new home, your body will be able to start making PAH enzymes that do what they're supposed to, process the Phe and protein, and keep your brain healthy. Gene therapy is full of possibilities. The National PKU Alliance is working with researchers and scientists around the world to bring us closer to a reality where PKU can be treated without a restrictive diet.
Now, let's move on to genetic engineering. Gene therapy involves altering the genes inside your body's cells in an effort to treat or stop the disease. It involves the removal of a desirable gene from a cell, adding it to the gene structure of a bacterial cell, and replacing the recombinant DNA into the bacterial cell. The bacteria then have the capability to produce the protein produced by the original animal or plant cell. To further understand about genetic engineering,
I made a video. These cats can glow in the dark. Now, is this a naturally occurring trait due to a random mutation? Nope. This is an example of genetic engineering, and we're gonna take a look at how this process works in this video. Note that genetic engineering goes by different name. You might also hear it called gen splicing, recombinant DNA, and genetic modification. But all of these terms mirror the same thing, which is basically this. Taking DNA from one organism, combining it with another organism, and getting recombinant DNA from two different sources. Now, in the case of our glow-in-the-dark cat, most of the DNA came from the cat, but it receives a gene from a jellyfish that allow it to glow in the dark. Now, you might be wondering how it's possible to combine DNA from a cat with a DNA from a jellyfish. Those are two different organisms. Well, the key is that the genetic code is universal. So if we were to take a look at the DNA of this animal, DNA of this bacteria, and the DNA of this little boy, we would see some differences, but they are all made up of A, T, C, and G. Now, the sequence might be different, but they're speaking the same language, and because the genetic code is universal, we can combine DNA from the different organisms, and genetic engineering is a reality. Genetic engineering relies on enzymes, two types in particular. So let's take a few minutes to see the role of these enzymes in genetic engineering. One of these enzymes can act like scissors. It's known as a restriction enzyme, and its job is to cut DNA in particular sequences. And then there's another enzyme known as ligase, and its job is to glue the DNA back together. Now let's explore restriction enzyme in a little bit detailed. There are many different restriction enzymes, thousands in each enzyme that is specific for a particular DNA sequence. So this restriction enzyme will only cut DNA when it sees the sequence CC, TGG. This enzyme will only cut DNA when it sees sequence TT, CG, AA. And this enzyme will only cut DNA when it sees sequence CC, TA, GG. The other thing to know about restriction enzymes is that many of them cut unevenly, and this is known as a restriction site. So let's take a closer look at how the restriction enzyme cuts unevenly. Here's the DNA sequence. Here is the restriction site for a particular enzyme. It likes to start cutting in here, but then finish cutting here. So what happens is that we get an uneven DNA sequence. These uneven DNA sequences are known as sticky ends. And the sticky ends because there are only one DNA sequence exposed, here it can easily bind through complementary base triangles to another sticky end. And this is important to genetic engineering. So here we've got a sticky ends. Now let's take a look at how we would use these two enzymes together to create a recombinant DNA. So here's our DNA sequence. Here's our restriction site for a particular restriction enzyme. First thing we're gonna do is add a restriction enzyme to cut it into fragments. In here you can see the sticky ends. Now we're going to add DNA from another source that's also been cut using the same enzyme. This is important because using the same enzyme means that you got the same sticky ends. So here is DNA from one organism, here's a DNA from a different one but same sticky ends. Now these fragments are gonna stick together because of complementary-based pairing. This sticky end was complementary to this sticky end, and this sticky end is complementary to this sticky end, so they naturally came together. However, we need a little ligase at this point because ligase will help attach the sugar phosphate backbones, which are more difficult bonds to make. So once ligase gets involved, here we go. We have our recombinant DNA, a DNA from one organism combined with DNA from another organism. So now let's take a look at whole genetic engineering protocol. and we're gonna use a particular example, a very commonly used example of genetic engineering. And this example involved the use of bacteria to the human insulin factories. That's right. Because of genetic engineering, we can make bacterial cells produce insulin. That's not bacterial insulin, it's human insulin. And then we can bottle that insulin and give it to the vets. So here's how it works. First of all, let's start with the bacterium. Why we will use the bacterium as a human insulin factory? Well, one reason has to do with the genetic structure. Bacteria have a big circular chromosome, but then it also has the smaller loops of DNA called plasmid. And it's very easy to move plasmids into and out of cells. So that's one advantage to using bacteria. The other advantage is that bacteria are unicellular, and then they produce asexual, which means that in a very short amount of time, you can get millions and millions of identical bacterial cells, clones. And this is important if you want factories. So let's see how the experiment works. We're gonna start with our E. coli bacterium. Here you can see is a chromosome, here you can see is plasmid. And then we get a human cell, and this human cell contains a nucleus which contains DNA, and within that DNA is a gene for producing insulin. This is the corrector gene that produces good insulin. So the first thing that we're gonna do is we're gonna take the plasmid out of the bacteria and we're gonna take the DNA out of the human cell. Easy enough. Then we're gonna cut both of them with the same restriction enzyme. So here the plasmid has been cut, and here the human DNA has been cut, and we're gonna use restriction enzyme that cuts at sites around the human insulin gene. So it's important to take the right restriction enzyme before your experiment. So now we have our human insulin gene and we got our cut plasmid. Now we're gonna combine those two together. Because of complementary base pairing and because of the sticky ends, they're going to naturally bond together. Then we're gonna throw in a little bit of ligase to help seal the sugar phosphate backbones, and now we have a recombinant DNA. Most of the plasmid is bacterial DNA, but it now has the human insulin gene. Step number 7. We're gonna stick this recombinant plasmid back into the bacterial cell, and this process is known as transformation. Whenever you give a cell new DNA, it's called transformation. So now we're gonna let this bacterial cell reproduce. And in a matter of days, we have millions and millions and millions of identical bacteria. All of them with this recombinant plasmid contains the gene for human insulin. So we're gonna let this bacteria produce human insulin, and then what we're gonna do is we're gonna take insulin out of the bacteria, and there is a special technique where we can isolate that protein. So now we've got the human insulin, and we can bottle this up and we can use it to treat diabetics who need doses of insulin. And then in a nutshell is how to do genetic engineering. Keep in mind that this is only one example. We can move genes from different organisms into other organisms. So one application is to make pesticide resistant in plants. These has a gene from bacteria that allows to produce their own pesticides, and we can make bacteria that can clean up toxic wastes. We can produce proteins to dissolve blood clots. We can make growth hormone, and we can even make acid wash genes.
Diseased people cured by fixing their genes. Putting genes into plants and domestic laboratory animals is one thing. What about using genetic engineering to modify humans, in particular to cure human disease? There are two quite different procedures that are being debated under this heading, and they need to be discussed separately. The first procedure is to insert foreign genetic material into the germ line into sperm or eggs where they could be passed on to succeeding generations. The second procedure is to use genetic engineering to insert foreign genes into somatic cells into any cells except sperm or eggs and their progenitors in an effort to correct some genetic defect. The role of genes in heredity. The heredity information is contained or within the genes located in the chromosomes of each cell. An inherited trait can be determined by one or by many genes, and a single gene can influence more than one trait. A human cell contains many thousands or of different genes in the nucleus. Heredity factors, known as genes, are thought to exist in discrete portions of chromosomes. The term discrete refers to the concept that genes are always located at the same point or on a chromosome. Genetic characteristics can be extremely complex and may require the actions of several separate genes to be expressed in the adult organism. An example of traits that require more than a single pair of genes to be expressed are human height and human color. Other traits may be controlled by a single pair of genes. Some of these traits may have extreme phenotypes such as albinism, a lack of skin pigment in humans. Now, we have common genetic disorder include Down syndrome, sickle cell disease, phenylketonuria, and hemophilia. Each of our gene codes contains unique protein, and each protein has a separate and distinct function to perform in the cell. To further understand, please watch this video.
Heredity, or why you look the way you do. Have you ever wondered why you look similar to your parents, but not exactly? Heredity is the passing of traits from one generation to another. But what are traits? Traits are characteristics that can be passed from one generation to another. For example, these may include your eye color, the color of a dog, the beak of a bird. In the 1800s, while growing pea plants, Gregor Mendel noticed that some traits, like the height of the plant, may change from one generation to the next. For example, if he bred two tall plants together, in the first generation, he would get tall plants. However, in the second generation, he may get 3 tall plants and 1 short plants. He classified the first generation trait as the dominant trait, and the trait that was masked or covered up as the recessive trait. The reason that you get dominant and recessive traits is that you get one copy of the trait from your mother and another from your father. Most human cells are considered diploid because they contain a set of chromosomes. This helps explain why you look similar to your mom and dad, but not exactly. Since Mendel's time, there has been a whole field of science that studies heredity called genetics. And lately, the field of genetics has been very busy. They have mapped the human genome, or the DNA, they have cloned animals, and they have helped to find cures to genetic diseases.
Now, let's move on to effects of genes in our environment. Genes are inherited, but their expression can be modified by interactions with the environment. Genetic traits are determined largely through the precise information found in the cell's gene structure. A variety of factors in the environment can actually alter the effects of a particular gene. Some examples of these effects are as follows. First, the effect of light or chlorophyll production. Second is the effect of temperature and hair color in the Himalayan hair. To further understand, please watch this video.
We face the challenge of feeding a growing population in a world with an increasingly unpredictable climate. Plants offer the potential to overcome this problem. There is a need to understand more about how plants respond to changes in their environment. We work to establish how plant growth and development are influenced by the environment, with a strong focus on temperature. Understanding the relationship between genes inside plants and how they respond to their surroundings will be an important step in sustainable food production. In this strategic programme, we have 130 scientists working on world-leading research to understand the mechanistic basis, regulation and evolution of genes in the environment. We are developing new technologies to assist research and breeding of important crops including brassicas and wheat. Interactions between fields across this programme, from mathematical modelling through to evolutionary genomics, will allow us to find answers to fundamental questions such as: how are ambient and seasonal temperature signals perceived, and how is the perception of temperature integrated into regulatory networks controlling growth, development, and defence responses? How do the layers of regulation generate the physical traits we see in the crops around us? How do genomes evolve in response to varied growing environments and adapt to duplication and hybridisation? Our findings can be translated into crops with larger, more plentiful fruits and seeds, oilseed rape with pods that resist shatter before harvest, plants with greater climate resilience. We have already established 2 lines of oilseed rape that offer reduced pod shatter, producing greater and more stable yields, and have developed the technology to fine-tune flowering time in broccoli, which would stabilise UK production, reduce food prices and food waste, and lower carbon emissions associated to brassica imports.
Now, let's talk about gene mutations. Genes are segments of DNA molecules. Any alteration of the DNA sequence is a mutation. Usually, an individual cell's altered gene will be passed on to every cell that develops from it. Gene mutations may be defined as being any changes in the nitrogenous base sequence of a molecule DNA. When the base sequence of DNA is altered, the amino acid sequence of the polypeptide for which it codes will likewise be altered. Such an alteration may affect the operation of the resulting enzyme, preventing it from properly catalyzing its reaction and thus preventing a trait from being expressed by the cell. The majority of gene mutations are harmful because they result in the cell being impaired from performing some specific task. In rare cases, a mutation may result in a lethal gene that kills the cell either by producing a substance toxic to the cell or by failing to produce a protein of vital importance to the cell. Gene mutations are passed to every cell that arises from the mutated. If the mutation occurs in somatic tissues, its defect is limited to the tissues immediately surrounding the mutated cell. If the mutation occurs in a primary sex cell, it passed on to the offspring that result from fertilization of or by game tests produced from the primary sex cell. On the other hand, DNA is a polymer made up of a repeating chemical unit known as the nucleotide. Thousands of the units are known to comprise a single DNA molecule, making it one of the largest of all organic compounds. DNA exists in hundreds of thousands, if not millions, of different forms depending on the precise arrangement of nucleotides in the molecule. Its variability is the key to genetic variation in living things. DNA nucleotides themselves are quite complex, being composed of 3 separate subunits: phosphate group, deoxyribose, and the nitrogenous base. Phosphate group is a chemical group made up of phosphorus and oxygen. Deoxyribose is a 5-carbon sugar made up of carbon, oxygen, and hydrogen. Nitrogenous base is a chemical unit composed of carbon, oxygen, hydrogen, and nitrogen. Bases found in DNA are adenine, thymine, cytosine, and guanine. To further understand, please watch this video.
Gene mutations are changes in the base sequence of the DNA in a gene. They can be brought about in several ways. Assume that the normal base sequence on a coding strand is ATG, CAT, GTA, and TTG. The amino acid sequence produced should be methionine, histidine, valine, and leucine. If one or more bases are deleted from the coding strand, the reading frame will be shifted. Thus, the amino acid sequence after the point of mutation will be altered. The resulting protein is usually non-functional. Similarly, if one or more bases are inserted into the coding strand, the reading frame will also be shifted, and the amino acid sequence after the point of mutation will be altered. the resulting protein is usually non-functional too. If a base on the coding strand is substituted with another base, a triplet code on the strand will be changed. This may result in one different amino acid in the polypeptide produced. If that amino acid alters the conformation of the protein sufficiently, the mutation would have an obvious effect on the phenotype. Similarly, if the order of bases on the coding strand is changed, a different amino acid in the polypeptide may also result.
Have you ever written a secret messages to one of your friends?
If so, you may have used a code to keep the message hidden. For instance, you may have replaced the letters of the word with numbers or separable following a particular set of rules. In order for your friends to understand the message, they would need to know the code and apply the same set of rules in reverse to decode it. Decoding messages is also a key step in gene expression, in which information from a gene is read out to build a protein. Genetic code allows DNA and RNA sequences to be decoded into amino acids of a protein. Let's move on to DNA code. The chemical and structural properties of DNA are the basis for how the genetic information that underlies heredity is both encoded in genes and replicated by means of template. As we recall that DNA is a complex organic molecule composed of thousands of repeating nucleotide molecules, and that each free nucleotide carries with it one of our nitrogenous bases. The particular sequence of nitrogenous bases adenine, thymine, cytosine, and guanine comprise a strand of DNA and providing the type of chemical code that is understood by the chemical mechanisms of the cell. The DNA code is used by these mechanisms to manufacture specific enzyme and other proteins through the process of protein synthesis. Each group of the three nitrogenous bases, known as a triplet codon, provides the information necessary to code for the insertion of a single specific amino acid into building protein molecule. The particular sequence of triplet codons on DNA enables amino acids to be linked together in a specific sequence during protein synthesis. In conclusion, DNA is not merely a molecule with a pattern, it is a code, a language, and information storage. This is how DNA code works. Please watch this video.
You may have heard of this expression that DNA codes for protein or for polypeptides, which are the building blocks of protein. And I want to show you in what language DNA codes for polypeptides and how that language is read. And so let's take a look at a chromosome. And if you were to take a closer look at a chromosome, you'd see that it's made up of a tremendous amount of DNA, all coiled up and wound up together. So we have DNA right here. And we're going to take a look at a very, very small segment of DNA that I kind of marked off with these lavender lines. And that would be a gene. And typically a gene codes for one polypeptide, for one chain of amino acids. And at some point, this gene will undergo transcription, and that means that an enzyme, RNA polymerase, will come along and make a strand of mRNA that's complementary to one of the DNA strands. And the DNA strand that it uses to make the mRNA is called the template strand. And the RNA polymerase doesn't pick the template strand arbitrarily. There are actually certain signals that will tell the RNA polymerase to pick one strand over the other, as one DNA strand over the other as a template, but we just won't get into those details right now. And so let's take a closer look at our DNA and the mRNA that we just made off of that DNA right here at the bottom left. And so you can see that all of the nucleotides on the mRNA are complementary to the nucleotides on the DNA, with the one caveat being that in RNA we have uracil, or U, instead of thymine. So wherever you would think to put a T, instead we put a U, which is uracil. And the mRNA will eventually land up in the ribosome, that's right here on the right, and the ribosome will translate the mRNA, and that means that it's going to kind of read it and put together the corresponding polypeptide chain. And so I just want to make it very clear that the DNA does not end up in the ribosome, it's only the mRNA strand that's read by the ribosome. And so every 3 nucleotides on the mRNA, and actually also on the DNA, is called a codon. And I'm just going to circle all the codons on the mRNA. So we have 1, 2, 3, 4, 5, 6, 7 codons on our mRNA, and actually also 7 codons on our DNA. And each codon is going to code for 1 amino acid. And let's see how that's done. So this chart that you're looking at, which is hopefully not too complicated, is the genetic code. It's the language in which amino acids are coded for and then put together into these long polypeptide chains by the ribosome. So in our upper right hand side, I actually redrew our original DNA and its corresponding mRNA, but remember, it's the mRNA that's read and not the DNA. And you can actually see that by looking at the genetic code. You can see that it's written in RNA language because there are U's and there are no thymines. So that kind of clues you in that it's written in RNA language. So let's actually look at the mRNA that we have and let's read it. Let's see what the ribosome would do if it encountered this exact piece of RNA. So we'll start with our first codon, AUG. So the way to read this chart is you start with the left side where it says first base in codon, and we look for the first base in our codon, which is A. And so we bracket that off and we're going to be looking at that row. Then for the second base, we look at the top of the chart where it says second base in codon and we find U. And here it is. We're going to bracket off this particular column. And then for the last base, we look at the right hand side of the chart where it says third base in codon, and we're looking at the part that was bracketed off by the row and column that we just looked at. So in this area, and we're going to look for G. And that's right here. We draw a line and we get the amino acid methionine. So AUG codes for methionine. And in fact, AUG doesn't just code for methionine, it also tells the ribosome to start translation. So I'm going to write start right over here. Let's look at our second codon, GUU. So we start with the left-hand side. We look for G. That's right over here. Then we look at the top of the chart and we look for U. So that is right over here. And then we look at the right-hand side and look for U in this particular area between those two lines. And that gives us the amino acid valine. So GUU codes for valine. Let's look at our next codon, CCC. So at the left side we find C, bracket off that row. Then we find C on top, bracket off that column, and then find C on the right. So CCC codes for proline. Let's do the next one, AAG. I'm going to go a bit faster here. So we find A, bracket it off, then look for A on top, and then look for G. It's a little bit hard to see under that line, but we get the amino acid lysine. So AAG codes for lysine. And the next one, GUU. So in fact, we already have the codon GUU. The second codon was GUU, so I'm not going to go over how to find it again. I'll just put in valine. Let's look at the next one, GUC. Let's find G right over here, and then U on top. So I'm going to put the bracket here, and then U. Sorry, C. We get valine. That's interesting. We have two different codons that are both coding for the amino acid valine. Let's do our last codon, UAA. So we find U, and then A on top, and then A on the right, and we get stop. So UAA does not code for an amino acid, but it tells the ribosome to stop translation. So there we go, we've synthesized our polypeptide chain. It's made up of methionine, then valine, proline, lysine, valine, and valine. And there are a couple of important things that you should keep in mind about the genetic code. The first is that it's universal. And what I mean by that is that all organisms, all the way from bacteria to plants to animals, all use the same genetic code to code for their amino acids and polypeptide chains. The second thing to keep in mind about the genetic code is that it's degenerate, which is just a fancy way of saying redundant. And that means that you can have more than one codon that code for the same amino acid. And we actually had an example of this in our polypeptide chain. We had two codons, GUU and GUC, that are different, but they both code for valine. So that's what we mean when we say that the genetic code is degenerate or redundant. And in fact, if you look at the chart, you can see that the genetic code is actually very redundant. There are many codons that code for the same amino acid. If you look right over here, for example, there are actually 6 different codons that all code for the amino acid leucine. And the third thing to keep in mind about the genetic code is that although it's degenerate, it is not ambiguous. And that means that each codon can only code for one amino acid. A codon can never code for 2 different amino acids. So if you look at the upper left-hand side, for example, let's look at the codon UUU. That codes for phenylalanine. So UUU always codes for phenylalanine. It will never ever code for a different amino acid.
Now, let's talk about the genetic code. Every organism requires a set of coded instructions for specifying its traits. For offspring to resemble their parents, information must be reliably transferred from one generation to the next. Heredity is the passage of these instructions from one generation to another. Discoveries in genetic science, deoxribonucleic acid or the DNA was revealed to be the chemically active agent of the gene. DNA replicates itself when chromosomes replicate in the early stages of cell division. DNA is passed from generation to generation during reproduction and acts as genetic factors. DNA interacts with the cell's chemical factory and produces the observable effects of the phenotype when genes are inherited by a cell or an organism. DNA regulates the production of enzymes in the cell and thereby enables the cell to perform the complex cellular chemical reactions necessary to sustain life. Stem cell therapy focuses on the use of stem cells to treat or prevent a disease or condition. Stem cells are a special breed of cells that are capable of differentiating into more than one kind of tissue. Stem cells are used for curing various diseases such as diabetes, arthritis, few cancers, bone marrow failure. Genetic engineering is a technique of controlled manipulation of genes to change the genetic makeup of cells and more genes across species boundaries.
Now let's talk about the importance of gene therapy in the future. Because of its accuracy, gene therapy has the potential to eliminate cancer cells without damaging normal, healthy tissue. Furthermore, cancer gene therapies may provide alternatives when a disease does not respond to other older treatments. Gene therapy is being used in many ways. Say for instance, replace missing or defective genes. Another, deliver genes that speed the destruction of cancer cells. Another one is supply genes that cause cancer cells to revert back to normal cells and deliver genes that stimulate the healing of damaged tissue. This is all for today, and I hope that you've learned something from my discussion. Thank you for listening, and goodbye everyone. For your assessment, please answer the following.